Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. My name is Aida Halidah binti Hamdan. So today I would like to present my research on the morphological characteristic and gamma irradiation effects on plant growth of Stevia rebaudiana. Okay. Now we proceed to the introduction. Okay. There are so many countries that are now commercially cultivating this Stevia plant. All major regulatory agencies around the world have approved or adopted high purity stevia glycoside as sweetener, and more than 150 countries has approved or adopted their use in food and beverage. Stevia has the sweetest flavor and contain other biochemicals that are beneficial to humans. It is brittle perennial herb with a large root system that produces thin elliptical leaf. Nowadays, cultivated plants are said to be more common. The leaves are the most useful component of this plant. But there's 18 species out of 110 tested for sweetness were found to have this trait. The seeds are extremely small. Since the seeds are so small, seedlings take a long time to grow. Infertile seeds are white in color, while fertile seed is black in color. The roots are thick and fibrous. Stevia is a short plant by nature, with a vital day duration of approximately 13 hours. Stevia will begin flowering once it has developed a minimum of 4 true leaves. And stevia also uh, known as they have very low rate of germination. Malaysia still lacks suitable varieties and production technologies, while a few countries such as Japan and India have successfully produced suitable stevia varieties. Therefore, there is need to carry out appropriate research and development program to develop suitable local varieties. Mutation breeding is one of the strategies that can be used in the agriculture sector to improve stevia genetics and variety. When opposed to traditional breeding methods, mutation breeding has the potential to broaden diversity and isolate desirable economic traits in a shorter period. Among other physical mutagenic agents, the most frequently used mutagen in mutation breeding are gamma rays. Okay, so this is my research objective. Firstly is to identify the radiosensitivity of stevia mutation after being exposed to gamma radiation. The second is to determine the M1 generation morphological characteristic of stevia mutation after gamma exposure. Okay, now we proceed to the materials and method. Okay. For the plant material preparation, the plant material was prepared by the stem cutting. Stem cutting method is the most easiest and the most cheap. It's a very cost effective. This method is used in order to grow the roots from the stevia. The second is the seeds of stevia were irradiated with gamma rays to induce mutation plant. Okay. So we irradiate the seeds by using the BioBeam GM 8000 Germany machine at Agency Nuclear Malaysia. So by using this machine, we can produce a mutant seed uh, uh, for the third is you grow the seed, germinate the seed by using the red light. Method is prepared by the Abdul Latif et al. With, and this method is allows germination under that environment. Next is we have to, after this is already germinate, we have to transfer the seedlings to the polybag and set up with the fertigation system. After that, the parameter were evaluated by qualitative analysis for the leaf. The quantitative analysis was measured on the plant height, leaf number, branch number, leaf width, and the leaf length. Lastly, the data was analyzed by using SPSS analysis. For the results and discussion, the sensitivity of potential mutant stevia. As we can see here, this is the table for the percentage of stevia mutation seed germination. Okay, we can see that the treatment seed from 100 gray record the highest rate of germination, meanwhile the lowest is from treatment 500 gray. Okay. So from the results above, we conclude that the rate of germination percentage decreases as the doses increase due to the negative effect from gamma radiation toward the stevia seed. Okay, so this is the survival rate of stevia seedlings. The survival rate is observed for 40 days after the seedlings is transferred to the polybag. Okay, so here we can see that the treatment for control recorded the highest, which is 75%. And the lowest one is from treatment 3 to 5 gray. And it is record uh, 0% survival rate for both 400 gray and 500 gray. Okay, this figure shows that the survival rate percentage to gamma rays dosage is formulated by y equals to negative 0.1436x plus 76.192, where y equal to 50. Therefore, we can get the LD50 by using this formula. And the LD50 is 0.82.40, which means that the 50% of the stevia seedling with that is exposed more than 182.40 rate of gamma dose. Okay. We proceed to the morphological evaluation on gamma irradiation treatment. Okay, so here we can see that the qualitative morphological characteristic from the leaf of stevia. Okay, so here there is variation of the leaf shape from the leaf tip, leaf base, and the leaf margin. Variation of shape could appear as different gamma dosage effect on the plant respectively. The gamma mutation disturbs the stevia plant stevia leaf characteristic at some point of growth stage. According to Izzati et al., the leaf characteristic was buried when treated with colchicine as the chemical mutation agent. And for this experiment, we use the physical mutation agent, which is gamma ray. There is also varieties that we can see from this table. So this is the picture from all the treatment. You can see that at 4F picture, there is abdominal leaf form. This is occur because of the gamma radiation itself. The gamma disturb the leaf formation, and an abnormal leaf form was observed. So this is the table for the growth parameter according to different gamma treatment dosage. This is by using the SPSS analysis. Okay, we proceed to the quantitative morphological characteristic of stevia treated with gamma rays. Okay, so this is the chart for the plant height. As you can see here that the treatment for 50 gray was the highest one, and meanwhile the treatment for 250 is the lowest. Most of the evaluated accession are different from each other morphologically, a good indicator of genetic divergence among all the treatment. As results, shown treatment 50 gray recorded was the highest in plant height among other treatment. Other than that, we can see treatment 0 gray, which is control, will also taller than the rest. Meanwhile, the lowest treatment in plant shows 250 gray. From the observation, the difference between the height of the stevia mutation could be due to the inhibition of DNA synthesis or other physiological damage after gamma irradiation treatment. Okay. Next is for the leaf number. You can see here that Treatment for control recorded the highest, which is 163, while the lowest one is 250 The numbers of leaf are widely used as an index in determining the biological effect of various physical mutations. As we can see, the number of leaf in control treatment shows the highest. Regarding the statistical analysis, there is significant difference in terms of the number of leaf. The genetic mutation interferes the biochemical process in the plant. Result plant height number of leaf are also important because leaf numbers is another valuable characteristic in stevia. Since the most glycoside content are found in the leaf, it is likely decrease in leaf number as the plant dosage increase. The next is the branches number. We can see here that the control is also the highest. Meanwhile, the lowest also from the treatment 250 The more branches present in the plant, the more number of leaves present. As we know, stevia is popular with sweetness compound. After 12 weeks, a significant difference on the number of branches was obtained between the gamma treatment. According to Majid et al., the number of branches were highly significantly reduced by radiationisis. It shows that maximum mean were observed 0 kiloradian as a control and minimum values for number of branches and number of leaves were recorded at 80 kiloradian. So we can see that the experiment also recorded the highest number of branches from the controls 0, 3. Meanwhile, as the treatment dosage increased, the number of branches also decreased. Okay, so this is the leaf width of the leaf. We can see that the control also recorded the highest, and the lowest is the treatment from 100 gray. Leaf width is another good characteristic to indicate larger leaf work, which implies to higher quantity of sweetness content in the leaf. Generally, the leaf is responsible for helping the yield quantity of stevia plant. In this study, the gamma irradiation did not affect the size of the leaf as the highest leaf ratio is from 0 grade stevia plant. For the leaf length, the control also record the highest one, while the lowest is from the treatment 250 grade. From the graph, we can identify that the treatment 0 has the highest leaf length. This is probably happened due to the alteration of stevia tissue, which caused the leaf to different dosage of gamma exposure effect in overall leaf size. The size of leaf plays a big role where a large surface area of leaf means that there are many chloroplasts to absorb sunlight. Chloroplast is the tiny structures in plant cell which contain chlorophyll, a green pigment that absorbs light energy for photosynthesis. Moreover, in stevia plant, the leaf contain chemical compound that make the leaf less sweet and useful for humans. The conclusion is the data presented has shown the presence of variability in the stevia qualitative and quantitative characteristics. The latter dose, also known as the LD50, for stevia mutant obtained from the study is 1 at 3 grade. This is probably due to the dosage of gamma rays radiation is too high, which causes the plant not able to show the good traits. This is also because of the gamma irradiation act as randomization during exposure to states with cesium-137. Hence, the possibility for further improvement using this variation is still wide. Thank you. That's all from me.